A patient in extremis presents with decerebrate posturing and a bilateral flapping hand tremor known as asterexis. Their central nervous system is rapidly shutting down. This massive neurological collapse is driven by a single blood parameter, a critical plasma ammonia panic value exceeding 150 micromoles per liter. A microscopic 17 gram per mole inorganic waste product, generated largely in the colon, possesses the structural capacity to completely dismantle the human brain. To understand how this happens, we must map the molecule's atomic architecture, trace its physiological sinks, outline disease pathology, and establish diagnostic stewardship. Before a clinician can treat a fatal encephalopathy, they must first trust the laboratory data. Yet ammonia remains one of the most fragile pre-analytical analytes in modern medicine. Ammonia's toxicity in the bloodstream is strictly governed by a dynamic pH-dependent equilibrium with a pKa of exactly 9.3. At pH 7.4, 98.5% of the molecule becomes the positively charged ammonium cation. Its symmetric tetrahedral geometry and 1.43 angstrom ionic radius perfectly mimic a vital potassium ion. This stolen identity allows ammonium to physically hijack the cell's electrical grid, slipping into sodium-potassium pumps undetected. The remaining 1.5% exists as the uncharged gas, ammonia. It features a trigonal pyramidal shape governed by a highly aggressive lone pair of electrons. Because this gaseous fraction is uncharged and lipophilic, it ignores lipid bilayers entirely, diffusing passively into central nervous system tissues. Total mass alone does not define ammonia's neurotoxicity. Instead, its specific geometry and pH-driven charge state dictate its infiltration pathways. Aquatic organisms can continuously dump raw ammonia gas directly into the surrounding ocean. Terrestrial mammals, however, lost this aquatic dilution matrix. We cannot excrete ammonia gas into the air without poisoning our own tissues. To survive on land, humans evolved the ornithine urea cycle. This containment system requires a massive energy budget, burning 3 distinct molecules of ATP to fix a single molecule of volatile nitrogen into stable, non-toxic urea. This exorbitant energetic cost buys a strict homeostatic threshold. It keeps the total circulating plasma ammonia pool in a healthy fasting adult rigidly suppressed between 11 and 35 micromoles per liter. Human survival on land requires dedicating a massive portion of our daily cellular energy strictly to pay protection money against our own internal waste. A common clinical assumption is that this nitrogen waste comes from somatic muscle breakdown. In reality, 70% of the internal ammonia load originates from colonic commensal bacteria fermenting dietary proteins using urease enzymes. This bacterial ammonia gas diffuses directly into the splanchnic portal circulation. This creates a continuous, highly toxic vascular river flowing straight from the intestines toward the liver. As this portal blood arrives, specialized periportal hepatocytes snatch up 95% of the incoming ammonia on the very first pass, feeding it immediately into the urea cycle. This hepatic filtration choke point operates at maximum physical capacity. It turns over the entire blood volume's ammonia pool every 2 to 5 minutes with absolute zero tolerance for downtime. The liver is the single, highly vulnerable shield standing between routine gut fermentation and total neurological collapse. In conditions like advanced cirrhosis, heavy hepatic scarring causes portal hypertension. To relieve the back pressure, the body constructs physical portosystemic shunts that bypass the failing liver completely. This adaptation mainlines the uncleared river of gut ammonia directly into the systemic arterial circulation. The lipophilic gas hits the blood-brain barrier and instantly diffuses into the neural tissue. Astrocytes, the caretaker cells of the brain, lack the urea cycle enzymes needed to process this gas. Instead, they use a desperate alternative, an enzyme called glutamine synthetase, which binds the toxic ammonia to a neurotransmitter called glutamate. This conversion produces L-glutamine.
While this neutralizes the ammonia, it introduces a fatal structural consequence. The hypermanufactured glutamine acts as a powerful organic osmolyte. This massive internal accumulation of glutamine forcibly draws massive amounts of surrounding water into the neural cells via osmosis. Because the brain is encased in a fixed, rigid skull, this uncontrollable cellular swelling quickly manifests as diffuse cerebral edema, spiking intracranial pressure and pushing the patient toward a fatal brainstem herniation. The brain's biochemical attempt to neutralize the ammonia is precisely the mechanism that causes the mechanical crushing of the neural tissue. This flowchart shows how consultants differentiate the root cause of an ammonia spike. By cross-referencing an elevated value with PT-INR panels and arterial pH, they distinguish primary liver failure from valproate-induced toxicity or inherited pediatric urea cycle defects.
The first-line chemical intervention relies on intestinal trapping. Clinicians administer lactulose, which gut bacteria ferment into acid, drastically lowering the colon's pH. This forces the uncharged ammonia gas to grab a hydrogen ion, converting it into a charged cation and permanently trapping it inside the bowel for excretion. For critical panic thresholds exceeding 200 micromoles per liter, chemical trapping is too slow. Clinicians deploy continuous veno-venous hemodiafiltration, a mechanical filter that physically strips the accumulating nitrogen from the blood pool. Managing hyperammonemia relies entirely on exploiting fundamental chemical gradients, manipulating environmental pH to corner and trap a shape-shifting molecule. To properly initiate these extreme interventions, medicine requires an exact measurement of the circulating poison. The analytical gold standard is automated enzymatic GLDH kinetic spectrophotometry. The machine combined blood plasma with alpha-ketoglutarate and an NADPH cofactor, initiating a specific enzymatic reaction. A spectrophotometer measures the exact rate at which light absorbance at 340 nanometers decreases, calculating the absolute mass of circulating ammonia based on how fast the NADPH cofactor disappears. The analytical accuracy of this automated platform is unquestionable. However, the machine is completely blind to how the blood specimen was handled before it was loaded into the analyzer. Blood samples drawn for ammonia are profoundly unstable because living red and white blood cells continue metabolizing inside the tube. This comparison graph shows the consequence of delayed separation. If a tube sits at room temperature for just 15 minutes, ex vivo enzymatic generation will falsely spike a healthy 30 micromole baseline directly into a 120 micromole panic zone. Another frequent pitfall is the fist clenching error. Applying a tourniquet and instructing a patient to vigorously pump their fist forces ischemic muscle to dump concentrated ammonia into the isolated venous pool before the draw. These basic bedside collection errors routinely generate phantom hyperammonemias. The false data perfectly mimics a comatose emergency, triggering massive, unwarranted alerts based on flawed logistics. To halt enzymatic activity, strict cold chain logistics are mandatory. The blood must be drawn into a pre-chilled green-top tube, plunged immediately into an ice water slush, and subjected to rapid chilled centrifugation. Diagnostic stewardship also applies to treatment. Ordering automated ammonia checks every 2 to 4 hours on a stable cirrhotic patient is redundant. The physical neurological recovery of the brain trails far behind the chemical clearance of the blood. Maintaining the human architecture requires the integration of several biological realities: the pH-dependent toxicity of the molecule, the continuous load from the gut microbiome, the efficiency of hepatic clearance, the astrocytic response, and the logistical rules of the laboratory. Ammonia sits at the lethal intersection of volatile molecular geometry and fragile logistical networks. Managing it demands rigorous, uncompromising consultant oversight to ensure the system holds.